Hello everyone. In this lecture, we look into amino acids, peptides, and protein, some of the basic properties and characteristic features. The main idea here is to understand and describe general structures of amino acids to gain some insights on their structure properties of common amino acids found in living organisms. We try to gain some in-depth knowledge on peptide bonds and their properties. We also try to study some of the selected methods of amino acid synthesis. And the ultimate idea is to gain some insights on primary, secondary, tertiary, and quaternary structures of peptides and proteins. It is good to have some thorough understanding of acid-base concepts, chemical properties of carboxylic acids and amines, stereochemistry, and knowledge about conformation of organic molecules. Let's start with the acids and bases. As all of you know, water is a weak acid and also weak base. The concentration of hydrogen ion and hydroxyl ion is 10 raised to minus 7 molar in pure water. We can therefore calculate the pH by using the equation, which is nothing but the negative logarithm of hydronium ion concentration, more precisely. Therefore, the pH of pure water is 7. And water also undergoes dissociation in the presence of weak acid, and through which it's possible to determine dissociation constant, which is called Ka. And when you have water in the presence of acid, it forms its conjugate acid here, which is called hydronium ion, and a conjugate base. Using the concentration of conjugate acid and conjugate base, we can calculate, and knowing the concentration of acid, we can calculate the dissociation constant and also the pKa, which is the negative logarithm of Ka. If you look into amino acids and their structure and properties, they all have something in common. That is, they have an amino group, they have a carboxylic acid group, and then R group. These are all connected to the carbon, which is called alpha carbon. And all amino acids have a primary amino group, and except protein, which has a secondary amino group. And all amino acids differ in their side chain R group. Based on the property of side chain, we can classify amino acids into polar, non-polar, charged, or uncharged. Amino acids play a central role in cellular metabolism. All organisms need to synthesize a majority of them. More than 500 amino acids in nature have been discovered to date. Their amino acids are different derivatives of amino acids, but only 22 of them participate in translation, that is protein biosynthesis. In 1943, Gordon, Martin, and Singe used a technique called partition chromatography to study and separate the constituent of protein that actually contributed to the rapid identification of 20 amino acids. By that time, that was only 20 were known to be encoded in the genetic code. Two additional amino acids, which were not used by all organisms, are identified as selenocysteine in 2000 and pyrrolysine 2 years after the discovery of selenocysteine. If you look into proteins, which are nothing but the polymeric structures of multiple amino acid residues, they're most abundant macromolecules in the living organisms. They vary in their size, structure, property, and function. And this variation actually defines their biological functions. All amino acids or proteins are end products of most of the genes. Majority of the proteins also undergo post-translational modification. That means once they're biosynthesized using the translation process, they can undergo some kind of substitution or derivatization, such as glycosylation. We can consider them as a polymer because it has multiple amino acid residues covalently linked to each other. They are linear and they are heteropolymer because more than one type of amino acids are present in majority of the proteins and polypeptides. The proteins can be fibrous or globular. There are 23,000 proteins in the human genome, and proteins constitute about 15% of the total mass of cells. If you look into the classification of amino acids, we can broadly classify them as nonpolar, polar, or charged, and not all of them fall into these classes. There are certain exceptions, such as histidine, which can be polar, but also it can be considered as a charged amino acid. There are some amino acids which can be both nonpolar and polar properties, such as glycine, tyrosine, and cysteine. Glycine forms a very simple example of an amino acid, and it is the backbone structure. And every amino acid has the same backbone structure except proline. And the only difference is the presence of R group attached to this alpha carbon. If you look into non-polar amino acids, they all have one property in common. They have certain aliphatic or aromatic chains which are attached to the alpha carbon, as you can see here. They only differ in the side chain, either aliphatic or aromatic or some heterocyclic rings. And proline is also known as non-polar amino acid, and also methionine belongs to this class. If you look into polar amino acids, they have something similar, but except the fact that they have some additional functional group. If you take out this hydroxyl group, this is same as a non-polar amino acid such as valine. And the functional group can be a hydroxyl group, or it can be a sulfidal group such as in cysteine, or it can be amides such as shown in asparagine here, or aromatic hydroxyl containing groups such as tyrosine. Serine, threonine, and cysteine are very important amino acids as they undergo a variety of enzymatic reactions. Cysteine actually forms a disulfide bridge, which is very important in controlling the stability of proteins and protein folding. As I mentioned in the previous slide, tyrosine and cysteine can also be classified as nonpolar amino acids. If you look into polar amino acids, they have some additional functional groups such as carboxylic acid groups or amines. This make them highly polar compared to simple uncharged amino acids such as serine, cysteine, or threonine. Because they present additional carboxylic acid group or amines, this can be fully or partially ionized at a neutral pH. Selenocysteine considered as the Nature's 21st amino acid, and the only difference here is that sulfur is replaced by selenium. So otherwise, all structure and components are similar to cysteine. It was discovered by Teresa Startman's group, and proteins that contain this selenocysteine are known as selenoproteins, and enzymes that contain cysteine residues are known as selenoenzymes. Majority of the selenoproteins contain a single selenocysteine residue. Selenocysteine is referred in a 3-letter code as SEC, and it has a lower pKa and reduction potential than cysteine. The 22nd amino acid is pyrrolysine. If you look carefully, it has a structure which is lysine here, which is covalently linked through an amide bond to 4-methyl 5-carboxylate pyrroline unit. The pyrroline unit, which is linked to covalently through this amide bond. It was discovered 2 years after selenocysteine. Initially, it was found in methanogenic archaea, but later it has been found in many bacteria. Cysteine containing proteins undergo very facile oxidation that leads to what we call as a disulfide bridge formation. And this is reversible if you provide some reducing environment, which is important as I mentioned previously in folding and stability of proteins. Disulfide bonds are unstable in certain cellular environment due to highly reducing nature of cytosol, for example, that therefore these disulfide bonds can be broken reversibly. Amino acids can be named and abbreviated in a variety of ways. If you want to look into polypeptides, a protein containing multiple amino acid residues, the residues are named by replacing INE with YL, like glycyl if you have 2 units, glycyl alanine if you have a peptide having glycine and alanine, and it is very simply by replacing this INE. We can also name them using a 3-letter code, and these 3-letter codes are the first 3 letters of an amino acid. However, there are exceptions if you look into many amino acids such as glutamine, aspargine. We have ASN, which doesn't really say the first 3 letters. That is because we have aspartate here, which takes the first 3 letters, and glutamate here that takes the first 3 letters. We can also represent them by 1-letter code, which is essentially the first letter of the amino acid. However, again, we will see many exceptions, such as phenylalanine, because P is already taken for proline, and we see Q, for example, in glutamine, and E and D for aspartate and glutamate. If you look into the composition and percentage occurrence, 50% of the amino acids are nonpolar, and only 24% of them are polar uncharged, and 25% to 26% of naturally occurring are polar charged amino acids. The amino acids here shown with the red font means they are essential amino acids. Amino acids have a unique property because if you look into the pKa of carboxylic acid groups, which lies from 1.8 to 2.6, and pKa of amino groups vary from 9 to 11. And pK of R groups also varies from 4 to 10, which is a large variation because it has R group can be carboxylic acid group or it can be amino groups. Since this variation in pKa between the carboxylic acid and amino group, a majority of amino acid exist as dipolar ions at pH, physiological pH, and these dipolar ions are known as Zwitterions. If you take a simple amino acid, you can easily change the charge by changing the pH. If you take polar charged amino acids, such as aspartic acid, then you can have a multiple charge. That charge can be cured by changing the pH, and that pH, which correspond to either below or above certain above or below the pKa of a given carboxylic acid group or amino group. In this case, we can have positive charge below pH 2 because both carboxylate will be protonated and also the amine is protonated. As we move towards pH 3, 2.8 below in this range, one of the carboxylate is protonated and the amine is still protonated. One other carboxylic acid group is deprotonated. When we are between 2.8 and 7, we can have a -1 charge because of 2 carboxylates are deprotonated. Above pH 10, all deprotonation occurs, even the proton from charged amine group. Therefore, it has -2 charge. The next important property of amino acids and peptides and proteins is isoelectric point. That means a pH at which a given amino acid has no net charge, it has a zero charge. If we look into this amino acid here, which is valine, we have a pKa of amino group, which is 9.69. pKa of carboxylic acid is 2.34. Now, to calculate the isoelectric point, which is very simple, you take the sum of these two pKa values, divide it by 2, then gives isoelectric point of valine is around 6.2. At this pH, valine is having zero net charge. What about if you look into multiple charged amino acid which are polar and charged, and how to calculate their isoelectric point? Here, in this case, if we take, there are two amino groups which have different pK values and a carboxylic acid group which has a pK 2.18. Now, in this scenario, you take the sum average of these two like charged or similarly charged functional groups. In this case, it's amine, so you make the sum of these two and then divide by 2. What if you have two carboxylates? Then you take the average pKa of carboxylates. So the pI in this case is 9.87. pI of aspartic acid is 3.22. That means at 9.87, lysine will have zero charge. At pH 3.22, aspartic acid will have zero charge.